All right, let's have a seat, please. All right, thanks for coming up so early. I apologize for not having coffee. That was a slip up in the schedule. But there will be, because I've already been up for 3 or 4 hours and I didn't think anybody needed coffee. But we will have coffee at the first coffee break, which is at 10 o'clock. And the coffee breaks will be in the cafeteria for those of you who were here last night. That's where we had the party. And we're also going to have lunch in there. So it's a buffet lunch. And it's free if you have your name tag. And the restrooms are that way as well. So just go across the breezeway and the restrooms are just on the left-hand side. There is no food or drinks allowed in the auditorium. You have your head chopped off, apparently. Anyway, without further ado, we have, just to remind those of people going to the Rusty Pelican, so that starts at 7 o'clock tonight. And the field trips start tomorrow, and you should have all been contacted by your field trip leaders for that. And then in the evening, we have the barbecue at the Coconut Grove Sailing Club. And there's nothing more to say. So without further ado, we have 11 speakers here, and we purposely chose nobody from the University of Miami or from Miami, but these are all basically colleagues of Bob and people who worked with him, as well as we couldn't get all old white guys, but we had to get some young people who worked with Bob recently. And so we sort of have, old, young, interspersed throughout the day. So that's the you can work out who you are, So we're going to start with, Bob's probably oldest colleague here. I hope I don't offend him by saying that. But Gene Shinn, and he's going to tell us it's all about the dust. And we all know that it's all about the dust, some of us, for a long time. But he's going to help us remember that. Gene. Of course, now we have to work out where your presentation is. Which one is yours? First one. What's it called? There's no name on it. Do you have your stick? Death in Florida Bay? There's one, the first one down. That one? Is it called Death in Florida Bay? Yes. Which one do I push for advancing? Oh, they didn't give you this one here. Okay, here, that works as well, but you can use this. You want to use that?
Yeah, I'll use it. Well, good morning. Had a lot of fun last night. I slept for about 2 hours. Worrying about this morning, of course. I'm going to tell you a story. This has not been published. And there were various political reasons why this didn't get published. You can figure out as you go. And I would like to mention that this got started with me back when I first went to work with Bob. I was put on 2 different projects helping people. And there were 2 main projects that I got involved in early on. I can push that button right there, right? Okay. And as you remember, I was a biologist at the time, and I was a lab technician, and so I wasn't a geologist. So a lot of what was going on was really new to me. And of course, you all know Bob was mainly interested in glorious mud. And so the project was trying to figure out the origin of the mud, and he had the theory, or hypothesis, that most of the mud in Florida Bay was being produced by penicillus and various algae like that make the little ragonite needles. So at the time, he was at cross purposes with Preston Cloud, who was convinced that the mud was being precipitated out of the water, as in whitings and things like that. Of course, I didn't know any better. And I was the technician. So the idea was to set up these little farms or little plots where we could measure the growth of penicillus. Well, the first thing I did was a big tour of Florida Bay. And you know, all of my diving and work had always been on the other side, out on the reefs. Florida Bay side, that was mosquitoes and horseflies and mangroves and nasty mud. So this was all new to me. And so I was to work with Ken Stockman, which we did. And so we set out these little plots that we would photograph every month and count the penicillus plants. And, you know, and then I would put them in Clorox and measure and did all this kind of stuff. And I think finally in the end, the conclusion was it would account for about a quarter of the mud in Florida Bay. That's assuming it wasn't getting washed out of the bay every time there was a storm, because we didn't know much about storms and what they did at that time. Bob was a uniformitarianist, and so it was all about day-to-day processes. And that all changed when we had Hurricane Donna, and I became a catastrophist. Well, as Malenbaugh called it, a catastrophic uniformitarian. And that was his phrase. So the thing that impressed me up in the northern part of Florida Bay, was the richness of turtle grass. The turtle grass was like that thick up in places like Whippoorwill Basin. And remember, when we mentioned Florida Bay, we were only talking about that area where all the mud banks are. It didn't include this big brown band that comes down here. And we didn't do anything there. I had no idea what was there. I know what's there now. But you notice the band goes all the way up to the mainland. And so obviously things grew very well up there. And we soon learned that there's not much water circulation up there. The tide is essentially nil in the middle, but you have tide on the west side and on the over here where you see all those mud banks. And I want you to notice those whitings there. Anyhow, that band now we know is actually pretty clear water. It's Syringodium grass, and farther north it's mainly turtle grass, you know, Thalassia. And so that was my introduction to Florida Bay. Well, in later years, during the early and mid-90s, all that grass up there started to die. And the question was, what was killing it? And well, I had an idea because I had already started getting interested in the effect of African dust. And I owe all of that to Nancy Maynard, who's here, who started our original funding to start working on dust. So I became a dust bunny, and so I blame everything on African dust now. So as you can see, there's Africa, and there's a big storm coming off of the northwest part of Africa. It goes all the way across. Now, I might mention that back in those days when we were working in Florida Bay, we didn't have any of these images. We had some black and white aerial photos. And so there's a lot we did not see. And here you can see it coming all the way across. And this is the stylized dust flow. You know, we have it in the Pacific coming from Gobi Desert area. And it comes all the way across the U.S. And then there's the dust from the Sahel. And I don't know if Joe Prospero's here, but this is Joe Prospero's data. Are you here, Joe? I remember when I first saw this, I really got excited. That big peak there in 1984, it was an El Nino year, but that's the dust in Barbados. That's the year the corals started dying all over the Caribbean. And I became convinced that something in the dust was killing the coral. And a little bit after that, I contacted Nancy Maynard, who worked on this back when she was here before she went with NASA. And she was starting a new project, and so we got some money to hire a microbiologist. She was aware that there were microbes in it, and I was concerned that there was as well. Joe Prospero wasn't, by the way. And so that started the dust project. Over there in 1973 is another peak. I remember being on a reef long before I knew about dust and wondering why I couldn't see the shore from land. So anyhow, I really got into the dust thing. And so I got suspicious when the central northern part of Florida Bay, turtle grass and sponges, just about everything was dying up there. And here's the dust records, you know, Everglades, Virgin Islands, Barbados, and the Canary Islands, of course, is closest to Africa. And then we started finding it in the laminated caliche crust. This is a place in the Keys. You see that layer on top, and that's a caliche soilstone crust, and it's red because of the iron in it. And it has quartz silt and things like that. And we eventually figured out that that's African dust. So it's been going on. If you take a core at a place like that, you'll find about 5 of these in the first 60 feet or so of the Pleistocene. But that's right on the surface. So it's been going on a long time. We did some further work. This is a small core from that crust nearby. And my goodness, it contains mercury. And you can see the carbon-14 dates. It's about 420 years at the top, and down a little over 3,000 lower down. And so that's the geological aspect of it. Okay. As part of a major project that actually was based on the idea that everything going wrong in the Keys was caused by big sugar. That's where the money was coming from to do the work. And this was a huge project, involved many universities and the USGS and the Park Service. All kinds of people were involved. And I found myself back out in Florida Bay, reliving my life, taking cores again because I knew my way around. Anyhow, this is a core from Bob Allen Keys, and it shows the the lead, when they started using lead, tetraethyl lead in leaded gasoline, and then you can see it drops out up near the top of the core when they took the lead out. There's also some coral data there, and lead was also showing up in the coral bands. Now, I think most of you will agree with me that that had to be coming from the atmosphere. Anyone disagree with that? Well, anyhow, that's what got me off on this crazy tangent. So, the idea was to sample surface mud. Now, Ellen Prager and Bob Halley had already done most of this work, and Ellen, remember that she ran these samples through the screens. We did the normal thing you do with screens, and she gave me the less than 62 micron, the mud fractions, the smallest stuff, because if there's going to be dust in there, it would be in the mud fraction. And then we needed more samples, so we went out and took another 65 samples. So there's something like 220 sample sites here. And so then they all went to a commercial lab for elemental analysis, and which is the point of this talk, by the way. And here's some more work, and this was done by Bob Halley and Mark Hansen and people at the USGS. They went all over the bay measuring salinity. Now, the high salinity in that blob there is because there's no tide in the middle of the bay. It just evaporates, and it'll range from being hypersaline to normal salinity. So that's the area of concern. If anything was going to accumulate there, fine-grained mud, I assumed that's where it should be. We also were doing groundwater, and this is a groundwater distribution, and you can see it gets pretty high up towards the upper part of the bay. And here is the percent mud, and I presume this must have been your work. Yeah. So you can see where the mud is. So, yeah, I think most of you agreed with me that the lead was coming from the atmosphere, and there's the lead distribution. You see it's right there, that red blob in the middle where there's very little tide, maybe that much tidal fluctuation. And also the lake areas between the mud banks, there's several feet of sediment. And the rest of the bay, it's basically bare rock, and all the mud is on the mud banks. The mercury, we didn't have as many samples for the mercury. You see, it's in a place over here near on the right-hand side. That's a small embayment that's cut off, and I assume that's where atmospheric stuff could accumulate. Now, there is mercury and African dust, and you also saw it in that crust that was several hundred years old and that I showed you earlier. So it's been coming over with the African dust long before, we had any industry. So it's not all from power plants, as you've always heard. Aluminosilicate clays, so these are clays. Now, it's pretty heavy over on the west side, but there's longshore drift coming down the west side. What did I do? Oh, iron. That's the red color that you saw. And most of the silica coming over has an iron coating on it. Phosphorus. There's quite a bit of phosphorus in the African dust, and again, it's in an area where there's very little tidal fluctuation. And copper. We found copper in the African dust, and you notice it's concentrated where in the middle and over in that little spot on the right that I mentioned. So, the conclusion that I made is that the death of Florida Bay in the mid-90s was due to African dust accumulating in this area where there's very little tidal fluctuation and where there's hypersalinity, et cetera, et cetera. Well, that didn't go over very well. The paradigm in which we were all operating was that it had something to do with big sugar, and there was a lot of money, so I decided not to publish it. So you're hearing this hypothesis here for the first time, and I'd be happy to discuss that with you, and I'm sure it's not the popular thing, but that's what I thought. So that's my story and how we got to where we are. If you have any questions, I'll try and answer them. Yes.
Gene, thank you for that. One of your charts, you showed that the lead was higher, or excuse me, the mercury was higher in the younger sediment. Is that correct?
Yes, the mercury was high in the upper part of the bay, and I can't really explain why it wasn't all concentrated in the middle of the bay. But of course, you remember, this was a time when you were told you shouldn't eat fish from this area because of mercury, and it was always assumed it came from power plants.
Well, I'm from the state of Michigan. We don't eat fish in Michigan. You're frankly suggesting that.
Yeah.
That's not from Africa, that is from
Yes. I'm not sure I heard all of the question, but we did a lot of additional work on the crust, even before we knew there was African dust in it. As you know, Gray Moulter did this work, and Ron Perkins worked on it, and Dan Robin published a paper, and he cut it apart like bacon strips and did carbon-14 dates. And there are places in the Keys where you get a contemporary date at the top and 3,000 or 4,000 years in just 1 inch. So it's when sea level was down and the dust is blowing over. Well, it's also on the Pleistocene limestone underlying the bay. That crust goes everywhere. And we mainly concentrated on the surface because Prospero's data would show that since 1965 was when we had an increased amount of dust coming over. And so we figured a place to look is at the surface. Plus, we had these samples that Ellen Prager had already taken. They were ready. And so we added to that. I don't know if I answered any questions. Any more? All right, well, I thank you, and you can either believe this or disbelieve it. That's up to you. That's why I didn't use a conclusion slide. I didn't want to be pinned down too much. But that's what I think, and every time I see something, I manage to blame it on dust.
Well, actually, Gene, we have a bit of time, so maybe you could tell us a little bit about the connection of dust and the death of corals.
Well, mainly it's correlation with that at peak time during 1983 and 84, and I realized that correlation is not confirmation, but it was a start. And then we did a lot of work on, individual bands and watching the dust increase. And, we had one student that even found the dust from Timbuktu, the volcanic dust, and corals from the Florida Keys. It would be good for someone to do these experiments. I've tried and tried to get this done. Now, we ran against the agendas of a lot of government agencies. I can tell you that the Marine Sanctuary Program didn't want to hear this. You have to think of the politics involved, and this is one of the things that's always bugged me about science, is that the politics get involved in everything. So Department of Agriculture was interested in that phosphate, but we couldn't get anything going. We worked with the EPA, and we told them the stories many times. Well, they were fighting industry all this time with their lawyers because they were blaming it on, smoke from power plants and so on. And it turned out that every agency that we approached, we ran into the same thing. Now, we finally got some major funding, and we switched horses a little bit, and a fellow named Dale Griffin is still working on this. The military is very interested. So we switched to the idea of bioterrorism, and we did find a lot of bacillus, various species of bacillus in the dust that you could culture. It's live. Did not find Bacillus anthracis, which is anthrax. But it's scary to think about that, but there's an awful lot of it out there, and we really don't know where it all is. And so Dale is still pursuing this, being funded by various military agents and involved with, the Desert Storm problem when all these people were getting sick over there. So, yeah, but that still doesn't prove that the dust killed the coral. Some experiments have been done by a high school girl that found that the dust killed the corals in her experiments. And you'll have to get Bill Precht, it's his daughter, he's here, have to get him to tell you more about it. But every concentration killed the corals in their experiments. And we don't know which one, what it was in there. We just don't know. And I also worked with a fellow that has an oceanography degree from this institution, who worked with the EPA, and he is, he's a fellow believer in all of this, that there's something in there, mainly pesticides. He did his dissertation on pesticides. They still use DDT and various pesticides in North Africa that are banned here in the U.S., and they are present in the dust. You name it, it's in there. And it's very difficult to get funding to, everybody we talk to gets excited about this, and the first thing they want to do is write a proposal to some agency to get funding to study this stuff. And I have to tell them, forget it, you're not going to get funding. But it is slowly catching on, and I think Peter Sport is, has a lot of interest, and one of his students here is interested. There are a few papers showing the relationship with red tide in the Gulf of Mexico, starts with the iron in the dust feeding the trichodesmium, and then that fixes nitrogen for the Carina brevis that causes a red tide. So there's many implications, and I would encourage anybody that wants to do this to do it, but you're just not going to get funded in a normal, because there are a lot of agencies out there that don't go along with this. So that's politics. It's one of my favorite subjects. Okay, Peter, I think I've dug a big enough hole for myself.
Right, well, we won't go into that. All right, thank you, Gene. Our next speaker is Erica Suosaari, and I'll just give you a little bit of introduction because she's actually a recent PhD student here at the University of Miami. She's obviously gone off and now she works for the Smithsonian, but she was a PhD student with Pam Reed, and Pam Reed was one of Bob Ginsburg's students, so she's really like a grandchild of Bob, you know. So that's her connection, and she did overlap when Bob was here and, you know, had discussions with him, and she's going to talk a little bit about her work on stromatolites.
Taking my thunder there. And which one's the pointer? So, the red one? Okay. And then this one, and then this advances the slides back, okay. All right, so good morning, everyone. So I was going to tell you that I was like the grandkid of you know, the PhD grandkid of Bob Ginsburg, but Peter just stole my thunder, so. I guess that makes Pam my PhD mom, and like Mitch, where's Mitch, my PhD uncle, and you know, it's the big Ginsburg family here. But I want to say thank you for the organizers for putting this together. This is a big room of carbonate aficionados that we get to spend the next few days together. I did overlap with Bob, and I was lucky enough to have him as a professor for one of the last classes he taught at Rasmus. It was Earth Surface Systems. And we went on field trips to see the Miami Oolite, to see the Florida mud banks, and to the Florida Reef Tract, where it was very treacherous and dangerous, and I wondered if I would get out alive and if he should be running these field trips. But we learned a lot, and it was a lot of fun. And I can also remember giving a presentation in this class, and having Bob doze off and thinking, this guy doesn't even care about what I'm saying here. And then, you know, I finished the presentation, and he's like, opens his eyes and asks the most detailed, pointed question about what I just said, and I learned very quickly not to, yeah. So Bob always paid attention. And then the other point is the big so what. So that was always his big question. And Bob did a lot with stromatolites over the course of his career and has influenced my career greatly, and I am very interested in that kind of environment and biology connection in the morphogenesis of stromatolites. And so today I want to kind of put those together in a tribute to Bob and examine stromatolites, so what, from a Ginsburgian perspective. And so first off, if we look at this kind of clock diagram of the Earth, you can see that stromatolites pop in here over 3.5 billion years ago. And as a modern geologist, we don't often think about the Precambrian. I'm very guilty of that. And it's an immensely long period of time. I mean, the Precambrian is this big brown area here. And this is some 4 billion years or more. In this time period, we saw the formation of the Earth as a planetary body, the formation of the geosphere, the formation of the atmosphere, the formation of the hydrosphere, and the appearance of the biosphere, and the transition of the Earth from a dead planet to a living one. And so from 3 billion years to 1 billion years ago, stromatolites are really, they dominate the fossil record. And you can see here, right before the Cambrian, they have this kind of pinch out. And this is because of the evolution of higher organisms that they either grazed upon the microbial mats or they outcompeted them for space. But you've got this really long time period before here where stromatolites really thrived in diversity and abundance. This is a mural from the Smithsonian where you can see this is depicting the early Archean, and you've got stromatolites all along the coast, some hot springs, volcanoes, and these are 1.8 billion year old stromatolites from Great Slave Lake in Canada. And so this period here was, I mean, this is most of Earth history here, dominated by stromatolites. And then this pinch out, they're not really that common today. And I think two of the best known localities for stromatolites are in Hamlin Pool in Western Australia, and also, so Hamlin, they were discovered in the 1950s. And then in the Bahamas, they were discovered in the 1980s. And so Bob had this long love affair with stromatolites. And he also pioneered comparative sedimentology, so that comparison of the modern to the ancient. And so this is Bob in the Bahamas here with this big old stromatolite. I think Noah was on this trip here collecting this. And then here you have Bob in Mauritania with Pam looking at fossil stromatolites here. But so he had this like long love affair. In 1964, he published a paper. It was a seminal paper on stromatolites, on the environmental controls of stromatolites. And he published that with Brian Logan, who did the pioneering work in Shark Bay, and also with Dick Rizak. Did I say that right, Benj? Rizak? Yeah, sort of. And that was called the classification and environmental significance of algostromatolites, so 64. Well, 27 years after that, he went back to explore the environmental controls of stromatolites and also the biological controls of stromatolites in a paper called The Controversies about Stromatolites, Vices, and Virtues. And here we are 27 years later, and I'm going to re-examine this paper. And so in this paper, Bob says, almost everything about stromatolites has been and remains to varying degrees controversial. Have these controversies influenced research and thinking about these laminated structures that play such a prominent role in Earth history? To seek answers to this question, it's necessary to examine some of the controversies and try to gauge their vices and virtues. So in this paper, he looks at the controversies, the reasons for the controversies, and the vices and virtues of those controversies. So the first controversy is the definition itself. Semi-Katoff had the most general definition, and it was great for identification in the field. But he didn't really make mention of the microorganisms in the definition. Now, these other definitions, Kalkowski, Walter, Crumbine, Schopp, they all said stromatolites were of microbial origin. Kalkowski and Walter didn't include lamination with their definition. Crumbine said they were laminated and the biology was interacting with the environment. And Schopp even brought it further and said that the stromatolites were specific to filamentous cyanobacteria and they were photoautotrophic. So the definitions run the gamut, capturing a purely descriptive definition at one end of the spectrum and an entirely genetic definition at the other. The second controversy, and this is my favorite controversy and what has definitely influenced my career, is are they fossils or are they sedimentary structures? And this is Bob's kind of nature versus nurture. And so here you can see as sedimentary structures, maybe there's some currents. Semi-Katoff looked at maybe some biostratigraphy. So are they biological fossils or can we use them to interpret the depositional environment? What Bob says is that the generally accepted definition or theory here is this trompette model, where the environment is the control at the macro scale and biology is the main influence at the micro scale. The third controversy is carbonate fixation. So precipitation of grains, are they made in situ or are they trapped and bound? And if you look at, there's a really big textural difference here. So these are ancient stromatolites, and this is a really fine-grained macritic laminations here, and then you get to the modern, and they're very grainy. So it raises the questions of what's the origin of micrite in the ancient stromatolites? Is the material alloxenous? Is it brought in, trapped and bound? Or is it autochthonous and created in situ? And then what accounts for the larger grain sizes in the modern? And so Ginsburg attributes these controversies to, first, the fundamental differences between modern and fossil examples, and two, the opposing philosophies between specialists. But mostly, the controversies are due to the inherent duality of stromatolites. And he suggests that the duality of the environment in biology is inherent in all three controversies. And there are vices and virtues to this. So the vices are that the polarization of the opinion regarding the duality detracts attention from its resolution, whereas the virtue is that the role of this duality is emphasized. Now, when I was invited to give this talk, I didn't really know what I was going to say or what I was going to have to add. And Pam suggested to me that I actually go back and read this 1991 paper. And I have read this paper many times. And in fact, we just recently had a paper published that deals with some of these vices and virtues in this duality. But I went back and I read this paper again, and I was just absolutely floored when I came across this statement here, which this is paraphrased. But so Ginsburg says in this paper, I rather expect that future efforts may well reveal that biology and environmental influences on stromatolite growth are closely coupled at the macro, meso, and micro scales. And so for the rest of my talk, I want to really focus on this duality and explore his hypothesis and kind of look at where the research has come from that paper 30 years ago until now, and look at environments in both Hamlin and in the Bahamas and the roles of biology and environment. But then I want to try to, hopefully, relate that back to these three controversies. And so first off, if we look at the macro scale, so this is, I should probably explain this too. This is the trompette diagram that I showed you before, so where environment's the main controller at the macro scale and biology is the main controller at the micro scale. This is that trompette diagram, but thrown on its side, basically, so it fits better on a slide. And so first we'll look at the macro scale. And to do this, I want to go to Hamlin Pool. And so Hamlin is where I've spent a lot of my career so far, my small career. I did my PhD research there, and I was hired by an organization afterwards and actually moved there and lived at Hamlin Station for a couple years. And so we've been doing field studies in Shark Bay from 2012 to they're still ongoing right now. And what we had in our minds when we went to Shark Bay was that Hamlin Pool is like, the stromatolites in Hamlin Pool were kind of like a ring around a bathtub. And that you would find the same stromatolites in the kind of the supertidal, and then you'd find the same stromatolites in the intertidal, and then you'd find the same stromatolites in the subtidal. And so we kind of went out with that idea in mind. But what we did, the majority of research that's been done at Hamlin has been done at Flagpole Landing and also up here at Karbala Point. And so we decided that we were going to go all over Hamlin. And what we found, so we took 45 stromatolite samples, and Harold Hudson's out here. I saw him earlier. He was out there helping us take stromatolites in 2012, I think. And we took pavements, sediments, microbial mats, water samples. I towed behind a boat for tens of kilometers, jumping off to do spot dives. We did our own single beam sonar survey, and we have a library of over 40,000 images, and most of them are geotagged. So we discovered that stromatolites are not all created equal, and I'll get into that a bit more later too. But so we took a new approach, which is to not look at stromatolites as this ring around the bathtub where you've got a certain stromatolite in the upper intertidal, and subtidal, but let's look at them differently from their morphologies. And so we had a number of different morphologies that were dominant in the pool, and then we also mapped these unlithified sheet mats and then subtidal pavements. And we put these together in a big map. And here down, this is Flagpole Landing, and this kind of area is a really well-studied area. And what you see, if anybody's familiar with Hamlin Pool, and the stromatolites there, oops. Well, here, I'm getting way ahead of myself. I don't know how to, okay, here we go. You see this kind of iconic stromatolite. This is what we think of when we think about Hamlin stromatolites. There are these columns, there's these domes, and that is what you would expect to find there, because that's an area that's been well studied. And up at Karbala Point, we have these subtidal stromatolites, these composite segmented stromatolites in the subtidal. But then that ring around the bathtub theory kind of falls apart when you get to other regions in Hamlin. Over here on the southwest margin, you've got these really long, linear stromatolites called Seif stromatolites that are the dominant facies type, lithofacies type. And up here, just south of the promontories in the subtidal, you get these elongate nested stromatolites. And in the northwest, you get these tabular stromatolites. And northeast, these elongate clusters. And in the south, you have stromatolites, but the area of stromatolites that you have is dwarfed in comparison to these big, really expansive sheet mats. And then up near the sill, it's a seagrass and carbonate sandbank. You don't get a lot of stromatolites. What you find are you have these boulders that are really macro-algal covered. And so if we look at representative transects from each kind of region, and you can see that there are remarkable differences between them, but there are also some similarities. And so we paired this with our single beam sonar survey, and we found that there are different regions. We split this. So this was the topic of my dissertation. We split Hamlin into these different provinces. And within these provinces, there are a lot of similarities in the shelf geomorphologies. But what we found is within those shelf geomorphologies, there are also similar associations of stromatolite morphologies. And so stromatolite morphologies are strongly correlated to the depth gradients, but then the energy of the system is strongly correlated to the depth gradients as well. And so you find that here, where we have in the south and also up in the northeast, we've got low gradients, we've got low energy, and we have very similar morphologies. So this unlithified sheet mats and then the elongate clusters. Whereas then in areas of higher energy, we find different stromatolite morphologies and associations. And so basically, morphology is linked to the gradients, which are linked to energy. And we can show this in different systems here. So that we have elongate nested stromatolites. They're actually quite similar to this. This is sandstone, actually, up in the Kimberleys in Western Australia. And you get these really very similar morphologies, but this is driven by energy. So when you have high energy, it's driving the morphology of the system. But now, over here on the right-hand side, these are elongate clustered stromatolites, and these are mussel beds at the same scale. And here, these are aerial photographs of both. So these are stromatolites, and these are mussels. Well, elongate clustered stromatolites are found in areas of low gradient and also areas of low energy. And these mussels, are actually self-organizing. And so in areas of low energy, you have stromatolites that also might be self-organizing. And so if we take this back to the controversy, well, first we take it back to the trompette model, where we've got environment being the dominant influence at the macro scale, and biology not being very influential at this scale. We can say that that's actually, I hope I've just shown that that's perhaps not the case. And that we have, when physical forces are really strong, we've got environment as the driver. But when physical forces are really weak, we might have microbes as the driver. And so if you bring that back to the controversy, which is Are stromatolites acting as fossils or are they acting as sedimentary structures? Really, they can act as both. So again, it depends on the surrounding environment. So at the macro scale, I think Ginsburg's prediction holds true. The biology and the environment are closely coupled. So now, if we go back to the trompet diagram and we look at this mesoscale, in the trompet diagram, environment and biology are supposed to have equal forces at this scale. And actually, our research has shown that it kind of really supports this. But what I want to do is go to that controversy and try to I'm going to try to tie it back into the controversy of definition, so trapping and binding versus genesis. And these are some of the 45 samples that we took in Hamlin Pool. And they're impregnated with epoxy, they're slabbed, they're polished. And one of the first things that I hope that you notice is that these internal fabrics of the stromatolites are really different. So some of them are really finely laminated, some of them have coarse lamination, some of them are clotted, and some of that is all happening within the same structure. And if we go to this original model of stromatolites, so this is modified from a Playford model in 1990, but these ideas go back all the way to the 60s, is that the microbial mat composition is determined by the environment. So you have pustular mats forming in the upper intertidal, and they're making an unlaminated fabric. You have smooth mats forming in the intertidal, and they're making a laminated fabric in the stromatolite, and coliform mats forming in the subtidal and making a moderately laminated stromatolite fabric. And how we've modified this diagram is we put these red boxes up here because the fabric of the stromatolite is a record of a former surface mat. So that mat is growing up through time, and it's leaving that record in the fabric. And if you look at this in another diagram, you can see that the pustular mat here, it's forming this clotted, kind of unlaminated texture, and that's have to do with the precipitation on the coccoid bacteria. But then you also have these other mats forming laminations, which has to do with some sort of recycling. And so if we go back to this diagram with trompet, we can say, yeah, that fabric is definitely caused by the environment and by biology. But if we bring it back to the controversy about definition, it's still really controversial. And if anything, the work that we did in Hamlin confirms that even more, because we can make assumptions about what the fabric will look like based on that microbial. Oops, sorry, here, I'm way ahead of myself. We can, the picture's gone, sorry. But we can make assumptions about what that interior of the stromatolite is going to look like based on where it formed in the tidal column. But like I said before and showed you in those pictures, I mean, you often have different fabrics forming within the same structure, and that's often a shallowing upward sequence because it's growing into a different zone in the water column. And sometimes we cut them open and there's these abrupt changes that lead us to ask questions about what actually happened to the relative water level to cause that change. And so you actually don't know what the structure is until you see the interior of it. And I think we can all agree that stromatolites are of biological origin. And so I'm on the side of wanting a kind of genetic definition for stromatolite, but I also want a generic genetic definition for stromatolite because we actually don't know what's on the inside of the structure until we see it. And so I think for the definition controversy, I prefer stromatolite to fall under the big umbrella of microbialite, but we can still argue that later. So moving on to the trompet diagram again, now at the microscale. So here at the microscale, we don't have a lot of influence from the environment, but we have a lot of biological influence. And I'm going to hope to try to relate that to the controversy of trapping and binding and precipitation. And to do this, I'm going to leave Hamlin and I want to go to the Bahamas. And this is work done from the Research Initiative on Bohemian Stromatolites. It was done from 1992 to 2012 and is still really ongoing. And this was spearheaded by Pam, and Miriam is also here. Miriam had a heck of a lot to do with that. And so at the microscale, we can relate those even to the mesoscale. And at the microscale, we need to think more about function. why are the laminations happening? What's the process here? And so if we look at this, this thin section made from a bit of stromatolite, we can see that there are trapped and bound sediments. That's this brown mottled bit throughout here. And you've got these blue lines. Here's a blue line in petrographic thin section here, and here's a blue line here. And these are micritic crusts, very, very similar to the micrite in Precambrian stromatolites. But then, oh shoot, then we also have these orange lines, which are micritized grains. That in here, they're kind of, you get these grain contacts where you get a little microbe called Silentium. It kind of moves through the grains in these contacts and bores behind it as it goes. And so you can really see the differences here. You've got trapped and bound grains, this micritic crust, and then the Silentia. And so what's the process that's causing this? And how do we move between the trapped and bound sediment, the microboring, and the micritic crust? Are they intrinsic factors? So is it ecological interactions, biogeochemical cycling, nutrient turnover, or are they extrinsic factors? Is it the environment? Is it hydrodynamics, sediment, light, water chemistry? Well, in order to figure this out, Pam and her team went to the Bahamas to watch stromatolites grow. And they did this in Highburn for many years. And here you can see that these bars down here are the changing of microbial communities on the stromatolite. And then above that, you've got burial and exposure. And then they coupled that with temperature, with light, with tide, with current, all sorts of environmental parameters, and tried to make sense of actually what's the process happening here. And what they found was that in order to make this lamination and precipitation, you've got a cycling of microbial communities. And that cycling of microbial communities happens because of these extrinsic pressures. So Schizothrix, which is the trapping and binding cyanobacteria, changes to a heterotrophic biofilm, so this micritic crust, as a result of changes in temperature and sediment abrasion on the surface. And another example would be Schizothrix, this Schizothrix community changing to not getting exposed to sediments and having changes in temperature, and then changing to a community dominated by diatoms, which may disrupt the laminations. And so environmental factors affect precipitation and trapping and binding. They affect the cycling of communities. It's temperature, light, wind, abrasion, exposure, covering. And observations from other sites also show that higher salinity leads to increased EPS production, which enhances micrite precipitation. So if we go back to the trompette model, we can see that you get this cycling of microbial communities that changes and causes changes in the lamination of the stromatolite as a result of these extrinsic pressures, whether it's water chemistry or physical parameters or sediment cover. And so now if we put all of these pieces together, if remember, this is the trompette model, the one that was accepted in the 90s. We have environment at the macro scale, biology is the driver at the micro scale. We can throw that out the window and say, this prophecy that Bob had in 1991, we can show that it's true, and we can back it up now with our current observations 30 years later, which is at the macro scale, you've got environment closely coupled with biology, also at the mesoscale and also at the microscale. So I hope I've done that a little bit of justice. So at the end here, to wrap up, stromatolites will probably be the focus of the rest of my career. And they're quintessential geomicrobiological products. So that stromatolites are an ecosystem in the palm of your hand. And that Ginsburg's prophecy in 91 is true. And the big so what here is now that we know that they're intimately linked at all levels, it's our challenge to deconstruct the dual influences of biology and the environment as recorded in stromatolites for 3.5 billion years of Earth history. And so I want to give a big thank you to Bob for influencing my career, and also to most of you out here in the crowd that I have directly or indirectly influenced my research interests and career. And I am really proud to be part of the Ginsburg legacy. So. Oh, do I have We've done a lot of water chemistry sampling, or done a lot of, so we know that we have salinities ranging from 18, which is less than normal marine salinity, parts per thousand, all the way up to 88 parts per thousand. Amanda's doing work right now to kind of carry on that with looking at alkalinity, and Amanda could probably answer this better than I could. But we have loggers out there and still have loggers out there, and we've done salinity, we've done the tides. I'm probably not answering your question here.
Mineral saturation of the water. I mean, your magnesium calcites and your alkalinity. Maybe Amanda and I should talk. One of the things you talked about when you go into the micro scale is how quickly you see that turnover. And there's a lot of study going on now in the micro sampling of you actually have a water chemistry layering on the micro scale. So I think you nailed it right on the head with kind of looking at this. And I was wondering if you guys started that work in Hamlin Pool.
We've started that work in Hamlin Pool, yes. That was Emily's diagram up there. That's a very pointed question for the Bahamas.
Also, it's being noticed quite a bit in low-custody environments. Utah, for instance,
Ah.
Salt Lake, they're starting to see the water chemistry change on a very rapid turnover with almost a weather event, and then that can affect the saturation. And right now it's undersaturated, but it can cause pockets of supersaturation that then does start to precipitate again. So I was just wondering if you're seeing any of that microscale
Well,
correlation yet.
we're still collecting water samples, so it's definitely an ongoing project. So I think we should all kind of link up and discuss maybe what kind of parameters we could look for.
That'd be great.
Anitra?
It seems to me that you might be well off to start collecting data, especially Shark Bay, on the color of the light, which is going to imply which microorganism, and particularly your sequence at depths, says to me, because there are various microorganisms that are responding with different productivity and different color lights. And as you're going down in Shark Bay, you're going from bright reds in the intertidal down to greens and blues through the yellows, depending on storms and other things. So that might be very interesting to go measure and try to interpret.
Definitely. Thank you. Yes.
Bill and then Jean.
About 30 years ago, John Grassinger published a zonation scheme for pre-Cambrian stromatolites up in the Great Slave Lake area. And it was very difficult to explain why there was such a clear zonation at a time when there were no biotic interactions, where you had definitely reef crest type stromatolites, shallow water tidal flat stromatolites, and then deeper water stromatolites. And this combination model explains almost exactly why you would have that zonation scheme.
That's Ginsburg 30 years ago.
Jean?
Yeah, when I was working with Bob Dill on the stromatolites at Woodstocking Island, we found they're like an artichoke. Actually, it was Bob Ginsburg who pointed that out. We originally thought they were little fingers, but they're like, and when you slice it, you get these things like this inside. And I didn't see any of that in the Shark Bay stromatolites.
They're really different systems in Shark Bay, and it's really interesting. And I had a lot of conversations with Ginsburg about that early on. I think we all did, Pam included, about kind of that diversity and the differences. And Bohemian stromatolites are really the environmental pressures that are on the Bohemian stromatolites and what allows them to form are really, it's quite different than the pressures that are on the Hamlin ones. And so I think that probably that's a result of that, of the interior fabrics that you do get.
Yeah, if Brian Logan were here, one thing he would point out is the importance of the late Holocene regression in Shark Bay. So the land is rising there relative to sea level, so most of the stromatolites did not form in the environment in which they now exist. Now, in his day, this was a total mystery, and I see you have it here also. But geophysicists now know exactly what's going on. There are two main reasons for it, and they're both related to the Ice Age. And so around the Laurentide and Scandinavian
Mitch and then Randy.
Sort of a mesoscale question. What causes this little ecosystem to produce laminated versus a clotted texture, so stromatolite versus stromatolite?
So in Hamlin specifically?
Anyway,
Well, I can talk to Hamlin. And I think we're still in the process of building that model for Hamlin, but it's definitely a differences in microbial communities. And it's been said that with, and I think it actually might be from a Walter paper, that or Flanagan, but the entophyllus, so that little coccoid that makes the pustular matter, that unlaminated, is that it's actually that the cement is forming around the coccoid, and so it's taking that shape and getting these little coccoid or the little clotted precipitates there. And with the laminations, they're different microbial communities, but also there's the cycling of either community or an environmental event that causes that. So we're still working that out. I imagine this will be the next. It was 20 years in the Bahamas.
Can I say something about
Yeah.
that? You can't make a laminated structure from a clunky mat.
Yeah.
If you have a clunky mat, you're going to make a clunky structure. If you have a flat mat,
I appreciate that. What I was getting after is the environment
Yeah.
coming into play.
Yeah.
So when you see that change in a single head, there's an environmental.
Yeah, definitely an environmental shift, which is driving a community shift.
And you know what environmental shift is?
Still working on that.
Randy, and then Tori.
Whether you're talking about the influence of now the sea level or like what are the ages of these stromatolites over which time then you would have to consider the processes that correlate to that duration. So the formol state is a very, very slow process, so it's hard to imagine that influence stromatolite unless the stromatolite is 1,000 years old or something.
Well, they are. So the stromatolites in Hamlin are about 2,000 years old, are the oldest age dates that we have. And they grow, so I think that the construction or the creation of a stromatolite is really kind of this delicate balance between what's precipitated and what gets held onto in the structure and what gets eroded. And so I know that some of the, like Phil Playford stuck some nails in years and years and years ago in Hamlin, and he could find, in some places up to 1 centimeter of growth in a year. And other places you get, if you kind of extrapolate that through age dating, you get about 1 millimeter per year. So it's, what's actually getting incorporated and what's not. And you do see this, and you do see this example of what Paul's talking about in Hamlin, where you get these really, so stromatolites probably formed a bit deeper, and then you get this pulse and this change of sea level, relative sea level, that then you see totally different structures forming. And some of them you'd expect to see this shallowing upward progression of what should be there, and then that's not there. It changes to a community that makes a fabric that you don't think should be there. And you've got this uplift that's happening there. And some of it is actually, there's some seismic activity there as well that's happening that's causing uplift. So, you see a bunch of different stuff. But, ultimately what I'd like to be able to do down the track is be able to kind of recreate exactly what the relative sea level has been over the last 2,000 years. And I think you can do that by reading the fabrics.
Well, right. I guess the shallowing upward is the concept of it is accreting vertically under a stable sea level. That's how you get the shallowing upward.
Yeah, yeah. But in some places you don't see that.
All right, we'll take 2 more questions for coffee. Tori, and then there was one at the back there.
Hi, yeah. Hi.
Hi.
What makes Shark Bay so conducive to stromatolite growth versus other compounds?
So Shark Bay is, it's an extreme environment. It's this restricted embayment. And the depth of the entire Hamlin Pool, the maximum depth is 11 meters. So you have these temperatures that range at 2 meters water depth. We have temperatures that range from 11 degrees during the austral winters up to 33 degrees in the austral summers. And I'm sure that it's even more extreme than that in the like right around the margins. But so you've got that happening. You've got salinity, you've got hypersalinity most of the time, so salinities that are double what normal marine seawater are, but you've also got this fluctuating salinity that's caused by a bit of probably groundwater intrusion, because you actually have less saline water in the summers when you'd expect there's no rain and high evaporation, and then during the winter you've got higher salinities when the water levels there shouldn't be as much evaporation. And so you've got that, but then you also have 2 meters of tidal change over the course of the year, and it's not really astronomical tide. You've got higher highs and higher lows during the summer periods. Am I telling this right? No. Higher highs and higher lows during the winter, and lower highs and lower lows during the summer. And you've got, so this is 2 meters over the course of the year, and you've got all these stromatolites that are just, right up close to shore. And so during one part of the year, they're mostly uncovered, and during another part of the year, they're covered by water. And so it's this really extreme environment. And we have a lot of questions about it, because you even have this little embayment right next door that's called Laherdon Bight, which has exactly the same environmental parameters, but there's no stromatolites. And so there's microbial mats there, and there's this monospecific shell of Phragma erigatum. And so it's really restricted, and that's the same shell that's in Hamlin. And I don't know what the dates are in Laherdon Bight for this shell of when the onset of it happened, but in Hamlin we know that about 5,000 years ago you start to see this monospecific shell, so you know that the environment got really restricted. So you don't see stromatolites then until about 2,000 years later. So why is that? Like, what changed in the water chemistry to get from this restricted environment to the growth of stromatolites? Or alternatively, is there a lag? And maybe the shells happen later in Laherdon Bight, and now, give it another 1,000 years and we'll start to get stromatolites in Laherdon Bight. So, I think I think people have always said it's the hypersalinity, but I actually think it's what Miriam likes to call the Goldilocks, is that there needs to be a number of factors that kind of all play into that to make the conditions just right for stromatolites to grow. Sorry, that was a bit rambly. I don't know, yeah. Well, but if you look at lake systems as well, there's a lot of small lake systems south of Perth in Western Australia that they don't have the tidal influence and they don't have other things coming, and you find a lot of clotted structures in there. And so, I don't know that I know the difference, but I think that's an interesting thought about them forming in deeper waters and what the reason for that is in stable environments. But then perhaps you don't get that cycling.
All right. Thanks, Erica. That was great. And now we have the long-awaited coffee, and it's in the cafeteria. Remember, no coffee in the auditorium, and we resume at 10:30.